Thank you, Vincent. And welcome all the panelists. Hi, Jack, you're back. Yeah, we can see Ken now. Rafal just joined, so I think we're all in here. So hello again. Now we're all the panelists are present. As Vincent said, we're going to have a short panel on the regulatory aspects of microbiome diagnostics. We have gone through a lot of the science over the talks today, and we're going to touch on a little bit on the regulatory aspects, which is also interesting. I guess probably Jack doesn't need a lot of introduction right now because he just spoke. I would ask Ken and Rafal to introduce themselves a little bit, just to set the scene.
Sure. Ken Blount, Chief Scientific Officer for Rebiotix, also VP Microbiome for Ferring Pharmaceuticals. We're developing microbiome therapeutics, finished a phase 3, have developed a lot of biomarkers along the way that we continue to pair with our clinical trials.
And I'm Rafal Iwasiow, VP of Innovation and Technology at DNA Genotek and Diversigen. These are companies of OraSure Technologies family, and where we at the DNA Genotek manufacture pre-analytic collection devices, stabilization devices, and Diversigen offers assays and services, CLIA-based microbiome services.
Thank you. Thank you both. So yeah, we want to keep this pretty open. So as you all have been doing quite actively during the talks, please feel free to type in your questions in the chat. I will be taking a look at them. Just as an introduction, so microbiome diagnostics don't have any particular or specific regulatory pathway compared to other kinds of diagnostics, which may be good or bad. Let's discuss this a little bit, but I would like to start off with a very generic question for all the panelists. What do you think are the main challenges of microbiome diagnostics? Is it the validation of biomarkers? Is it any robustness of results, reproducibility? We shouldn't just a warning. We shouldn't get too much into the biomarkers because there will be a full panel on biomarkers and diagnostics tomorrow, but yeah, probably that word comes up a couple of times. But just to crop your early thoughts on what are the major challenges about the development or the commercialization of microbiome-based diagnostics. Maybe we can start with, I don't know, Jack. Yep.
Okay, I'm happy to start. I've already made the point, right, that I think that time is important. Hopefully everyone saw that. But in my research life, because remember I'm a research professor, not a co-founder of the company, but I'm not. I'm on their SAB now. But that's the primary driver for identification of biomarkers is that high dimensionality over time, which creates this problem of biomarker observation, right? And so really digging into when those biomarkers are present and what that presence actually means functionally for host microbiome dynamics is the key component in my mind. And then obviously the resolution of identifying what biomarker it is, right? E. coli is not just E. coli, it can be up to 600,000 different strains, some with up to 60% of their genome different. And if Lewis, if I took your genome and I swapped out 60% of the genetic material, you'd become a fungus. So that's an E. coli is not an E. coli. So we being able to identify exactly what we're referring to, which I think Rebiotix has been very good in identifying biomarkers which are particularly associated with that, and lots of other companies have, is key, right? Key for regulatory purposes, and obviously key for actually functional bioinformatics.
Yeah, I would add to that and say I think one of the challenges is simply the newness of the microbiome field. It's not as though we're familiar with the biomarkers like we would have been with blood pressure or prostate specific antigen. It's not like the biological association of these biomarkers has existed for 10 years. So as we're defining, much of what you saw from Jack today was cutting edge biology of understanding a certain state and how it associates. We just simply don't have those associations. And therefore, it's hard to go to a regulatory agency and say, look at all this body of work in the literature that makes it clear that that biomarker correlates with a clinical outcome or a clinical condition. And that's the way they think about it, is a large body of work that's very convincing that it would be a biomarker that they would consider qualified or validatable.
And maybe if I could add to that, what I'm hearing both of you say is it's around standardization, right? So those bodies of work and the changes that we're observing, right? I think one of the challenges of interpretation of that from a regulatory body or whoever else is the different methodologies that are used, right? So standardization of those methods end to end to avoid those changes that were observed, to understanding the new technologies that are being employed as well. I think is certainly a challenge. And I think, then if you go even further, I think it's the clinical utility, right? It is the challenge today. I think we're doing a lot of great science, right? And, driving towards that clinical utility, but I don't think it has fully been communicated to the regulatory bodies, which part of the clinical utility we're talking about yet.
And the regulatory agencies in our experience do think a lot about what you just said there with the standardization. They also think about, what are you trying to use it for? Which, maybe we get into that as well. Are you trying to identify patients? Are you trying to use it as a surrogate endpoint? And their view of what's required is very different depending on what you're using it for. And also depending on whether you intend for it to be a broadly accessible public biomarker or something specific to your regulatory program. They view that very differently.
Do you think it is just a matter of time that we collectively, as an industry or as a human race, generate more data that may lead to higher significance? Or do you think the lack of standardization will be a real challenge even in the long run? Or are you seeing alternatively, convergence towards standardization, or you are identifying that each one is doing their own thing?
It's inevitable. I mean, I love a quote. It's the sound of inevitability within the industry that we're going to have them. It's just a matter of time. There's too many companies, too many researchers that are working towards this. Just the natural progression of science will inevitably get us there.
Yeah, I mean, the key thing here is, yes, a vast quantity of data is great, but as Ken was pointing out, never before in the history of medicine really have we tried to deal with something, maybe in the brain, have we tried to deal with something that's this high dimensionality and this high level of dynamism, and create biomarkers from it. I mean, I'm hammered that point home, but it's a key point, right? And this is why when companies in the past, some of whom are no longer in existence, have tried and to go with consumer-facing products to identify these things, I've railed against them, because I've always said, well, hey, not to blow my own horn, but if I can't do what you're saying you can do, how the hell do you think you can do it, right? And that's always key. I look at these organizations and I go, I'm sorry, I don't think that's technically feasible. And a lot of the time it wasn't, because as Ken pointed out, or Raphael pointed out, we just don't have the data over this time frame to answer that question. It's just a ridiculous statement. And this is still going on. There are still a lot of companies out there that are really pushing for this. And I think that's damaging in my mind to a certain extent, to push it forward. But it is, of course, it's inevitable. As Ken points out, it's essential that we generate this data, but also there's because the microbiome isn't just blood pressure and it isn't just linked to CVD, right? It could be associated with a cornucopia of chemistry. To perform an iteration this early in the morning can be difficult, that can influence so many different facets of human physiology. How many clinical trials do we need, and how many different disease states or health traits to be actually able to identify biomarkers that are of relevance to this number of variants? We could need more data than we've needed for virtually any other disease state in the history of medicine. And that produces a profound problem, right? You need better standardization, as I keep on hammering home, and more data than we've required before.
Yeah, and I want to touch on a couple of facets you just mentioned that I think are very important. The multidimensionality. This is not data that you can say, all right, let's go run a Fisher's exact on. There's to put it kindly, there's a lot of hacks out there that do statistical comparisons with microbiome that simply are not correct math. It's multidimensional, it's compositional. You have to use very specialized statistical tools to really correctly compare compositions. And so that means that the tools to develop these biomarkers are also developing along with the body of data. And all that means, and can sound very daunting to say, oh my gosh, it's going to be 30 years before we ever get there. And that's the conundrum. The conundrum is we simply don't have the luxury as an industry or as a healthcare system to wait for the same level or the same perspective of evidence. We need to be creative about the ways we demonstrate evidence to the point of saying, yes, this is a useful biomarker. We can't think traditionally, and this is something that we've tried to have a discussion with the agency about, is we've got to be more creative. We got to think creative the same way that people urge the agency to think creatively about small orphan populations and not doing the same large clinical trials. This is what we need to do with microbiome biomarkers, and we are trying to have those discussions.
Yeah, I love that point, because that's key, right? My colleague Rob Knight is probably the behemoth in this field for statistical generation of bioinformatic tools associated with microbiome analysis. And in the NIH understands that, but when we talk to the FDA, they're still hung up on clinically mandated statistical approaches, which just don't really work. They break when we're dealing with microbiome data. And so that imagination, a lot of which is still in Rob's head, and Rob's an SAB on the SAB for BioSense, and is building a lot of the backbone statistical framework for dealing with this. Because, for example, BioSense, we producing now tens of thousands of metagenomes. If I gave you 1 million metagenomes and said, and analyze it, that's not a trivial problem. And anybody that tells me that's a trivial problem doesn't understand what they're talking about. I guarantee it. That kind of thing requires new statistics, as well as new computational approaches, right, to handle the dimensionality, to do dimensional reduction where necessary without losing signal. So those components are incredibly important to understand.
I think those complexities are important to understand from a regulatory framework, right? I think there's a probably longer path to helping the agency and other regulatory bodies understand what that means, right? But I think there's probably easier paths in the short term, where some of these complexities get distilled to, a subset of organisms, right, within the microbiome. And that is a way of establishing potentially some early clinical utility for the microbiome as a whole, right? And I think it's going to be a step-by-step kind of process to gain acceptance of the microbiome. I know at Diversigen, we not only do metagenomic sequencing, but equally we do qPCR for specific targets, right? And that's all about, once you identify those through research and the right statistics, or identify those biomarkers, trying to establish, some kind of ground truth that, regulatory bodies will be able to accept and understand.
Yeah, translating that to metabolomics, right? We go in and we need a particular metabolic characterization pathway for saying, okay, I found this metabolite, it's generated by these microbes, great. Okay, well, let me have a particular chemical reaction that allows me to detect it reliably and reproducibly in the human blood. It's the same as a qPCR approach, right? And that becomes absolutely essential.
So mentions to the regulatory agencies have been, common in your discussion. My follow-on question actually was, and the first question that we got from the chat is, do we need, or do we think we need a new regulatory approach, a specific regulatory approach for microbiome-centric diagnostics? Or is it more about, the industry doing their homework and, scientifically and also educating authorities on how these correlate or can be parallel to regular or other diagnostics. The point was, do they present any particular regulatory differences because other complex diagnostics tools have been approved, like based on gene expression, multiplex microbiological testing, and all that. My personal opinion is that I don't think we need any specific regulatory framework. Maybe this is a difference with pharmaceuticals, in which some specificities may be required for the approval of LVPs or fecal transplant. My personal opinion is that we do not need, but I would like to hear your thoughts.
Yeah, I'm going to answer that with a yes and a yes. Yes, we do need a different framework, and yes, we need more dialogue with the agency and with the community. So currently, if you need to think about how we talk about biomarkers. When you talk about something that the entire community, the healthcare system and companies would use, a single biomarker, that's really much more like the FDA's biomarker qualification program. That's currently regulated out of the Centers for Drug Evaluation and Research, which is primarily focused on biologics and small molecules, not Centers for Biologics Evaluation and Research that handles microbiome. So you've kind of got two different parts of the agency, one that normally thinks about microbiome, but they're not the ones being tasked at this point with microbiome biomarker development. So I think there is somewhat of a new model when we're talking about something generally accessible. The second half of that is how to approach that and how to have those discussions. I think the community engaging with the agency is the way to get there. I think it's a collective education and a collective work of the community with the agency. So I'll sort of tell you what we're doing as a company and as an industry. So we're part of what's called the Microbiome Therapeutics Innovation Group, or MTIG, which is a consortium of companies that directly interact with the agency on several issues. So we've had these discussions about biomarkers, and last year we had a meeting with the FDA in which we said, hey, why don't we have a workshop where we discuss novel ways to think about regulating biomarkers. So we're working towards doing that and pulling in a broad swath of the community to do so. We've been somewhat delayed because that's the type of workshop we really want to have in person. Turns out we have a pandemic, so that's a little tougher. But I think this is the tool by which you start to have these thoughtful discussions about how to develop these biomarkers that could be used in general for the entire community. So that's the route, and at least the way I see it.
I think Rafal was saying, though, and I also agree with this, that there are low-hanging fruit, right? There are elements which can be retrofitted into the existing approvals framework for particular biomarkers. The small molecules are key to that, and I mentioned metabolomics. BioVentures' platform is being adapted to do metabolomics, so we can preserve chemical signatures effectively on the device. And that will be, in my mind, the key game changer for identifying chemical signatures, which are essentially small molecules, which can be used as a biomarker akin to what's already been done for 30, 40 years in pharmaceutical identification of those small molecule biomarkers. And that will enable rapid retrofitting into a broad array of approaches, right? Microbial ecology, right? Ecology is probably a dirty word in the pharmacological side, but that's essentially what microbiome science is. It's microbial ecology, right? Is still long-haired people with beards like me who go out and poke around in holes, and it's still messy because ecology is always going to be messy, right? And so I think there's definitely low-hanging fruit which can be built into the new program, but as Ken says, that education is absolutely essential. The community needs to tell the regulatory agencies exactly how to standardize the analysis and interpretation of this data.
Yeah, I completely agree. I mean, just from our personal experience at DNA Genotek, we've done this on the genomic side as well. So for over 10 years, we've been engaging with the FDA, the agency, and other regulatory bodies. We've taken devices through that didn't have a framework, and it wasn't clear how we would go through it, but it was all about dialogue, early engagement, a lot of discussions, right? So that education is very key. And from those examples, I can tell you, we carved out a new path to approval for these kind of devices, right? I suspect the same thing will happen in microbiome. So engagement discussions are key early. Maybe I disagree a little bit with Ken that we need a different framework. I think what we need is different guidance and different approaches within the regulatory agencies, right? But I think the general framework still holds. All the regulatory bodies, use a risk-based framework to gain approval, right? And it will come down. I'm not sure it was Jack or Ken mentioned in the previous question. Like, what is the clinical utility, right? There's different uses for the microbiome, right? And some will be much lower risk, maybe around monitoring or than diagnostics, right? If I use those two as a comparison, right? So if you go for the lower risk one, I think it's a easier path, right? And if it's a diagnostic, I'm sure there's going to be guidance and more scrutiny around the body of data that's going to be required to get those approvals.
Yeah, and I think that's an important point. That's sort of what I was angling at with what are you going to use it for? For instance, if I want to use a biomarker to select patients for my trial, that's relatively low risk to the patient. And the FDA is perfectly willing to give you all the rope you want to hang yourself with, as long as it doesn't hurt the patient. If it's something that's really intended to determine a clinical response, that's a much higher hurdle, because is that clinical response that you measure with a biomarker truly indicative of clinical benefit? And I think that's really the perspective that the agency takes, is protecting the patient, ensuring the benefit to the patient is their first and foremost concern appropriately. I wanted to touch on something you said, Jack. I love that you put in the ecology. I think as an industry, we're trying to fit, unfortunately, some of us are trying to fit into a conventional box of drug development. This is ecology, plain and simple. I mean, it's the ecology, stupid, to use to bastardize a quote. I mean, that's really what it comes down to, is an ecology. A good example is I saw a publication earlier this year about a microbiome pharmacokinetics in drug development, and it's just flat out wrong. We don't know what that means as an agency. As an industry, what does pharmacokinetics for microbiome therapeutic mean? And we're trying to force it into that box. And in fact, when we've sat down with the agency and asked these questions directly, what role does pharmacokinetics play in a drug approval of a live biotherapeutic? Their hands go up and they say, that's not required for your approval, because guess what? We don't know what that means, and the industry doesn't know what that means. So I think that's a great perspective to say, yeah, we can fit into some of those boxes. Pharmacodynamics for a microbiome is a little easier to imagine, because then you're measuring something that happened in the patient. But pharmacokinetics, I still have not seen any convincing model that says this is pharmacokinetics for a drug. And I think we're somewhat remiss trying to force that fit. So ecology, love it.
I think we all agree on that.
I'm going to get t-shirts. Ken, I'm going to get t-shirts. It's ecology, stupid. I love that. That's perfect.
I think we'll agree on that. I'm not sure, however. I think I'm more of the opinion of Rafal. And I want to open up this discussion. We don't have a lot of time, and we have a lot of questions in the chat. But is it that it will be tough to get a diagnostic tool approved in any jurisdiction, and focusing more on the US where you guys are based? Is it that it will be tough to get it approved completely from scratch, or is it that it will be difficult to get the 510K on the diagnostic? So sort of these, substantial equivalence. So is it that it will be very difficult to show substantial equivalence with anything that is in place, even if it is PCR based or whatever other means? Or, in short, the question is, how difficult do you think it will be for microbiome diagnostics developers to get approved via the 510K?
I mean, my opinion is that it's really going to come down to what's the risk and benefit to the patient, to be honest with you. I think to get something broadly accessible that selects patients that would go through a 510K is very different than to get something that is determined as a clinical outcome or a trigger for reimbursement in the healthcare system. All of these are going to have a higher bar. Those that end up with a meaningful decision made from the biomarker.
It always comes down to the proposed indications for use, right? So, you know, the more complex those are, the harder the path. But I think as you know, the leaders in this area, as they carve out these paths, it's going to get easier and easier, right? So with each engagement with the agency or any regulatory bodies, as we figure out how to navigate here, it's going to lay a path for the next one to come through and follow those guidance, right? So it's not easy for sure, and it's a long path, but I think there is a path to getting there.
Yeah, and it also depends upon the populations that are being analysed, right? You know, we're trying to push heavily upon improving the diversity of cohorts, for example, that are being assessed. A biomarker identified in a group of 300 people from one country of one race or, you know, ethnicity is you know, ineffectual if it hasn't been applied to a much broader and more diverse cohort that's of relevance to the disease burden. And so, you know, and that key component comes in when we're trying to identify what are the strategic approaches that need to be developed in order to actually get a particular biomarker approved. And a lot of companies don't think about that, right? You know, they think, oh, that looks like interesting IP. I'll license that, or I'll develop it independently, and we'll go from there without thinking about, well, how relevant is that? And a lot of that, again, comes back to the dimensionality, standardization of approaches, types of protocols that are being leveraged to generate this data.
Jack, I think you raised a good point there. You know, from a microbiome, you know, I've heard people talk about a core microbiome. Like, what are we comparing it to, right? You know, we talk about dysbiosis. What's the definition of dysbiosis, right?
But do not ever get me started on dysbiosis. I will go mental.
But I think those are the kind of things that when you think through, you know, the regulatory approach and how you're going to take it through, those are the things you need to think about, right? What is it that you're trying to establish? And when we talk about, you know, unfortunately, the word microbiome encompassing everything, it's much more difficult than thinking about a biomarker or something that you can really point to.
Let me just riff on dysbiosis for 2 seconds, Ken. It's funny.
Say something similar.
When I hear dysbiosis, I hear I don't know what's going on, right? It's you know, ecology stupid, but dysbiosis means I don't know what I'm doing, right?
It's not dysbiosis, it's stupid.
Yeah, exactly. Yeah, it's not dysbiosis stupid. I mean, you know, there's a wonderful definition. Mariana Bindlos, who's a fantastic early career scientist, came up with a much better definition, and it's all mechanistic, right? It's saying that there's a problem here, and this is dysbiosis. This is this disconnection between the microbiome and the host, right? When it's no longer a productive symbiotic relationship, but it's now broken down, right? So define it mechanistically, like you said, Rafal, and I'm with you. Just say, you know, you've got low diversity. If we've got dysbiosis, I'll come after you with a pitchfork.
Yeah, well, and I think there's not a universal dysbiosis anyway. I think it will end up being intratypes of dysbiosis for this disease state, for that disease state, and maybe even a couple of different interotypes within a disease state, right? We've seen that for IBS, certainly there are multiple interotypes, even if there is a clear dysbiosis associated with IBS. So I would agree, you can't just say dysbiosis. I mean, what do you mean, right? It's broken. That's all. I don't know. I have no idea where it's broken or how it's broken or what it means, but it looks like it's broken to me. Yeah, that's kind of what
the mechanic saying to you, yeah, car's broken. Well, can you fix it? Well, I don't know really. It's just broken, you know. Okay, thank you. I'll buy a new car.
Rubbish.
Okay, so we're of time. We're actually a little bit over. There are a lot of questions. I think what we can do is, you know, each of the panelists maybe can take a look at the other questions in the chat. A lot of them, a couple of them on computer tools and artificial intelligence that may be interesting as well, a couple of them. So otherwise, I think we should call it a panel for now. there's a small short break of 23 minutes right now, and we will be resuming in 23 minutes. So thank you very much to all the panelists. I hope that everyone enjoyed, and see you very soon.
Great, thank you.
Thank you.
See you now.